four types are acute lymphocytic leukemia, chronic lymphocytic leukemia, acute myeloid leukemia, and chronic myeloid leukemia. Then the last type, we usually call that as lymphoma. Lymphoma, from the name you can understand, lymph is there, lymphoma. It begins mostly in lymphatic systems, and there usually it's unusual type of a cancer, lymphoma. And there are two types of lymphomas are called Hodgkin lymphoma and non-Hodgkin lymphoma. Okay, so now coming how cancer spreads. So as we know that as cancerous tumor grows, bloodstream or lymphatic systems may carry these cancer cells to other parts of the body. And during this process, cancer cells grow and develop into a new tumor. This phenomena, we call it, when this occurs, when mostly cancerous tumors spread, or it carry cancer cells through bloodstream or lymphatic system to other parts of the body, and then they are uh, and after after carrying that to different parts of the, or to some specific parts of the body, it usually the cancer cells, after transportation, the cancer cells grow and they may develop into new tumor. This whole condition we call is as metastasis. Okay, then one of the first place, what happens, a cancer usually often spread to lymph nodes. And what are these lymph nodes? Lymph nodes usually play a major role in the immunity of our body, in the immune system of our body. Why? Because these lymph nodes are the organs, are one of the major organs of our of our immune system, that helps us to fight infection in our body. Okay, so in the first place, a cancer may often spread to mostly in our lymph nodes, and it can also spread through bloodstream to distinct parts of the body. And these parts may include the parts that where it is carried away, where it is transported. These parts are include mostly bones, liver, lungs, or brain. Even if cancer spreads, it is still named for the area where it began. If after spreading of cancer to different parts, the cancer is still named for area where it begins, where it originates, not to the part where it is transported. As for example, if breast cancer is spreading to our lungs, then we definitely don't call it as lung cancer. We will call it as metastasis breast cancer, not lung cancer. So this is one of the another important characteristics, that uh, where the cancer is named according to its origin of development, not to its not to the to according to the name where it is transported. It is not. It is only named after where it has been originated as a uncontrollable or due to uncontrollable cell division. Then in cancer, we will again come to some different types of terms, which is known as, one is known as neoplasm. What is neoplasm? Neoplasm. So neoplasm is also known to us as an abnormal growth of cells in the body. And this neoplasm can be a small growth, such as mole, that can be developed in our body. Mole usually is a type of a cell where cells are not uh, differentiated into distinct type of cells. So our mole that is present in our body, that can be represented as an undifferentiated mass of cells. So we can call that mole as neoplasm. Or this neoplasm can be cancerous, or maybe or it may be represented as a precancerous tumor. And neoplasms are not dangerous like cancer cells. Okay, this is most important. So neoplasms are the examples of neoplasms, that some abnormal growth that occurs in the occurs in some specific organs in our body, such as adenoma is an example of one type of neoplasm. Fibroadenoma, these are the examples of neoplasm. And this abnormal mass of tissue that develops in neoplasm, that usually forms when cell grows and divide more than they should, or do not die when they should. That is why we get the ultimate result as neoplasm. So we get neoplasm as abnormal mass of tissue that forms when cell usually grows and divide more than they should be, or they do not die when they should. Okay, that's why the result we get as neoplasm. And mostly, if we differentiate between neoplasm and tumor, what is the main differentiation? uh, we refer tumor to as swelling or lump like swollen state that would normally be associated with inflammation. Inflammation is a term that is used mostly in immunology. Inflammation is usually a result of some abnormal reaction to the cell. There are four steps of inflammation that that is accompanied with inflammation, some redness, some swelling. These are some of the steps that are included in the process of inflammation. But so tumor always, this is an abnormal growth, or a lump of cell that is grown to a swollen state, and these always associated with inflammation in case of tumor. Whereas in neoplasm refers to any new growth, lesion, or ulcer that is abnormal, that should not be associated with inflammation. So both tumor and neoplasm, we call it as abnormal cell growth, whether they are malignant or benign. But in case of tumor, other things with immune systems are related, but in neoplasm, they are not associated with some events that are associated with immunology. And tumor mostly, it's an excessive cell growth or division in our body, and mostly it causes tumor occurs due to already mentioned, due to interference in the genetic changes, genetic factors. And that genetic factors include many things that I already, mutation, transversion, deletion in DNA. And other factors are also related with this formation of tumor. That is, if we are exposed to UV rays, if we are exposed to some radiation, some chemicals, some smoking, some tobacco, if we are exposed to tobacco, lung cancer is the best example. If we smoke, if some people are a chain smoker, ultimately the lungs gets affected, then lung cancer can take place. COPD can take place. After that, it can develop maybe to lung cancer. Then viruses are also agents which are the common causes for the development of tumors in our body. Some Epstein virus. These There are some special types of virus that can also be the cause of the development of tumors in our body. And mostly in the process of formation of cancer, that is cancer is the process of formation of cancer takes place by 4 steps, and these 4 steps are mostly tumor initiation. First step is tumor initiation, then the second step is tumor promotion, third step maybe it is converted to malignant conversion, and the fourth step is tumor progression. This is the fourth step that is involved in the process of carcinogenesis, that is the development of cancer. Now I will be showing you the slide that will cover almost mostly the details of your cancer in your syllabus. So is it the slides are visible?
Yes, ma'am.
Yes, ma'am.
Okay, so coming to cancer and carcinogens and the process of carcinogenesis. So this is how, if we see a close look, a cancerous cells look like, where you can see that these are the normal size of the cell, and if when it is divided uncontrollably, how it looks like, you can definitely differentiate between the size of the normal and the abnormal cell division growth from here. So coming to the cellular basis of cancer, what are the cellular basis of cancer, how cancer can develop? So it's usually an uncontrolled and inappropriate division of cells. Then the second reason can be genetic damage to critical genes that regulate cell division, cell growth. Then the third one can be exposure to chemical carcinogens, which are considered very important. If you are exposed to something carcinogen, some chemicals, or that chemicals may include many things. There are lots of things around us. We are living in an environment where there are lots of chemicals, lots of substances that are not useful for our body, that are considered as carcinogens. Carcinogens is is mostly defined as agents which cause cancer. So these chemicals may include pesticides, insecticides, which are very much nowadays days applied to for a beautiful growth of fruits, or some attractive looking fruits. People are giving the farmers, not the farmers actually, some companies which are growing those types of agricultural products for their sell, for their benefit. They are using pesticides, insecticides, so that they have a good variety of agricultural products. Recently I was going through an article where it is said that in China, Chinese people are using a lots of carcinogens in some agricultural products, mainly in fruits like apples, like many other fruits that fruits that are developed in China. Also, the rice noodles which we are consuming, they also give some chemicals in that rice noodles also. Green tea, rice noodles, all the things that are coming from China, they are tested in the labs of US, and also they are tested where they are in, where the agriculture products are imported from China. In also in Germany, in Australia, they have tested, and they have found that most of the agricultural products that are imported from China, they consists of chemical carcinogens. and that are very harmful for our body. That is why nowadays cancer is in so much, what I say, in so much, it's so much developing in not only in our country, in western country also. Some in specific types of cancer are developing very much due to these carcinogens, due to these chemical compounds which are harmful for our body. Our body cannot metabolize that harmful chemicals. That is why they acts as an agent which helps us to for our body to develop abnormal amounts of cancer cells. So so then coming to then how this occurs, then we are coming to that. If we see in a normal cell division, what we see that a mother cell that divides into two daughter cell, but after formation of that daughter cell, if maybe one daughter cell is okay and the another daughter cell where a damage can occur, and if no repair occurs, then no repair can occurs, many things can happen. Cell can go to apoptosis or cell suicide occurs, but that is the normal process. So we can see here that apoptosis is a process which can prevent the cells from going in from developing into cancer. Okay, but in case of cancer cell division, what happens, we will see that in case of cancer, the cell division, in if there is some if there is no repair system works in that cell, no apoptosis occurs, then what happens, then this cell, the first round of mutation occurs, then the rate of mutation, see in the second mutation the rate of damage increases, then in the third mutation, the cell ultimately more and more damage occurs, which is known as third mutation, and in fourth or latter mutation, what we will see that cells totally get uncontrolled, and ultimately we what we get as cancer is like this uncontrolled growth, which is beyond our control, beyond the control of a system of our body. So this is what we get as uncontrolled growth. So in normal case, what we will see in normal cells, we will see that our cells can go oxidative stress. There may be some interaction with carcinogens. There may be some interactions with viruses. And after all these interactions, what we get, we will get DNA, our DNA damaged DNA, and also there is some special type of alterations that can happen in our body, which is called epigenetic alteration. And what is this epigenetic alteration? Epigenesis is the process where there are changes, but there alterations can happen, but without the change of the DNA sequence. So that can be called as epigenesis. Other alterations can happen, but the DNA sequence will remain intact. But in mutation, the DNA sequence will be changed. This is the difference between epigenesis and mutation. So after that, what happens then? If there is a modulation of cell fate, what can happen? Then the potential causes, if the cell is modulated, what is that modulations? The modulations can be some potential causes, that is, if it is failed to repair, the cells are failed to repair, mutations in regulatory or repair genes, then failure of the cell cycle checkpoint mechanism, activation of growth promoting genes, inactivation of tumor suppression genes, and inactivation of apoptosis regulatory genes. all these, if all these incidents occur, then ultimately we will get as cancer. Okay. Then if there is successful DNA repair, if after if apoptosis occur, if the cell cycle arrest DNA occur, if DNA can get repaired, then ultimately we will get back to that normal cell. But those if those things do not occur, then we will get as cancer. Then development of cancer. Then this development of cancer, there are many points which can help in the formation of cancer. One can be the unlimited growth. Another point can be, if the cell during its cell cycle, if they ignore the checkpoints, or they can, if they turn off the tumor suppressor genes, and the tumor suppressor gene is p53. Yesterday I told, this is also called the guardian of the genome, because um, it's mostly protect the cells, it arrests cell division. So that is why this is called as tumor suppressor gene. So that is one of the points. Other points are, if the cell escape apoptosis, if there is immortality in the cell. Immortality means there are unlimited divisions in the cell, where where the chromosome maintenance genes can be turned on. Then there may be then there may be promotion of cell blood vessels growth. And this blood vessel growth, formation of many blood vessels within our body, is known as angiogenesis. Angio means blood, genesis means blood vessels, angio means, and genesis means growing off. So if we have, due to abnormal growth, many many blood vessels can also grow in our body. This is an accessory part of a cancer development, that is angiogenesis. So here, that is another point that can promote development of cancer. Then there are some also incidents where a cell can overcome anchor and density dependence. Means they are inside our body, within some organs, there are some many anchorage dependent cells. That is, the cells has to be anchored there in that fixed position. If that is disturbed, if anything is disturbed, the density and the anchorage of the cell is not correct, then cancer can also develop. Okay. So these are the 6 points which promotes the development of cancer. We can also call these as 6 key hits of the cell that can promote cancer. And hits can, and the mutation, that is the changes that are occurring in the cell, that is also referred to as hits. Okay. Then coming to the special terms that are used in the area of cancer, or oncogenesis, you can say. The main terms are, what is cancer, what is tumor, what is neoplasm, what is neoplasia, what is benign tumor, what is malignant tumor, what is metastasis, and what is carcinogenesis. These are the special terms. These are only used in oncology or in cancer. Okay. So cancer, what is that? It's a malignant tumor. It's not malignant, it's not the right word to use in the definition of cancer. A tumor, you can say, that has the ability to metastasize or invade into surrounding tissue. Tumor, a general term for an uncontrolled growth of cells that becomes progressively worse with time. tumors may be benign or malignant. Neoplasm, same as tumor. Neoplasia, the growth of new tissue with abnormal and unregulated cell proliferation. Benign tumor, a tumor that does not metastasize or invade surrounding tissue. Malignant tumor, a tumor that has the ability to metastasize or invade into surrounding tissue. And metastasis, ability to establish secondary tumor growth at a new location away from the original site. Carcinogenesis, the production of carcinoma, epithelial cancer. Sometimes carcinogenesis is used as a general term for the production of any type of tumor. Then coming to mutation. What is mutation? That is related to genetics mostly. Mutation is a sudden heritable change in genetic material or character of an organism. Okay, how this occurs? You see, this is our DNA. These are the genes, the and these are consists of the purine, pyrimidine, and everything, the nucleotide sequence and everything. And if it is this DNA is exposed to some incoming radiation like UV rays or photon or anything, then what happened? After that, we will get that some of the DNA nucleotide sequence can get disrupted, or it can be changed to other nucleotide sequence, or in, and that leads to mutation. Okay, and what is epigenetics? Epigenetics are all the genetic modifications, including changes in the actual DNA, excluding changes in actual DNA sequence. That is called epigenetics. And these modifications, including adding molecules like methyl groups to the DNA, which is called as methylation. If some modification occurs in the nucleotide sequence or in DNA, and that modification can be some adding of molecules like methyl group, CH3 group to the DNA, which is called methylation. Acetylation can be another type of modification. And due to these changes, the appearance and structure of DNA, which alters how that gene can interact with the transcribing molecules in the cell nucleus. That is, we call as the whole phenomena we call as epigenetics. These are the special terms that you will get familiarized when you study genetics. But since cancer is related to the interference of some genetic changes, that is why these terms are coming in the chapter of the cancer. Okay. So what causes those hits? Hits means the mutations. So mutations in cells are usually triggered by this chemical and physical agents. What are those? Can be UV radiation, cigarette smoke, pollution, age, genetics, chemical exposure, radiation exposure, and heat. Then there are lots of theory associated with this cancer. Some of the important theory are such as example is two-hit theory of cancer causation. In this two-hit theory, it is mostly related with the genetics concept. Usually, in the what is told in this two-hit theory of cancer, usually we know that normal cells have two undamaged chromosomes. Normally, we know that we have two undamaged chromosomes. One is inherited from our mother and one is inherited from our father. And this chromosome contains, we know, thousands of genes. So, and if some people with their hereditary susceptibility of cancer, if cancer is in their heredity. Then what happens? People with heredity susceptibility to cancer, they can inherit a damaged gene on one of their chromosome. So their first hit or mutation occurs at the conception. Though in those family, in those people's family who have a who have that incidence of cancer in their family, in the first, when conception occurs during the birth, they get that first hit or mutation in one of their chromosomes. And what then, after at their birth they get that one hit or one mutation, what happens? Other people may receive the first, maybe in later stage, or later, what I say, later in their uh, inheritance of their family. Maybe other people may receive that first hit at the later stage, before or after birth. In that case, we call this theory as two hit hypothesis. One hit, normal cell, one is heated, then after that some mutant gene is also acting. After that mutant gene is acting, then we will get that two hit cell. One is the one before, one at the time of conception we get one hit, and after, in the later stages of the life, we get another mutation in their gene. That is considered, that two is considered as two hit cell. So ultimately uh, what we get, we call this situation as two hit hypothesis. And this two hit hypothesis is given by a scientist called Knudsen's. So we call this as Knudsen's two-hit hypothesis. This is one of the hypothesis, or this is one of the theory of formation of cancer causation, or formation of cancer. And this theory, Knudsen's two-hit hypothesis theory, is one of the important theory. Okay, and usually this theory is dealt with one important gene, that is called retinoblastoma gene. Okay, and a mutation in retinoblastoma gene is shown here, which is usually a very rare event to occur. Mostly in retinoblastoma gene causes eye cancer retinoblastoma. And in the childhood, this eye cancer retinoblastoma, people who inherit the gene, and people who inherit the first hit are 100 times more likely to develop the second cancer causing mutation. Okay, the people who inherit this gene, retinoblastoma gene, at the first hit, or at the time of their conception, they are most likely to cause second cancer causing mutation. That is why it is called two hit. One hit occurs at the time of birth and the next hit at the later stage, so it is called two hit. Then coming to hallmarks of cancer, means what are the most common factors that causes cancer, that is termed as the hallmarks of cancer. So cancer can develop due to many factors. These are the factors given here. Due to some cell growth inhibitors, which are known as EGFR inhibitors, epidermal growth factor receptors inhibitors are there, which the inhibitors, they sustain the proliferative signal. They sustaining that or inhibiting that proliferating signals that are present for the cell, that are needed for the cell for their proliferation. That can one reason. Some cyclin dependent kinase inhibitors are there, Yesterday we have studied in cell cycle. There are maybe some cyclin dependent kinase inhibitors which can inhibit some growth, which can inhibit growth of the cell, evading the growth suppressor genes that can cause cancer. Some immune cells can be there, that is called immune activating. Some antibodies can be there that can avoid the immune destruction and leads the cell to cancer. There may be some telomerase inhibitors are there that that have the capacity of replicative immortality that leads to cancer. There are some anti-inflammatory drugs present that can lead to tumor promoting inflammation which ultimately causes cancer. There can be some inhibitors of some signal transduction process that can cause cancer, and these inhibitors ultimately activating inhibition and activates metastasis. There can be some inhibitors of angiogenesis, means some inhibitors uh, which helps in the formation of blood vessels growth that can lead to cancer. Some PARP inhibitors that can cause genome instability and mutation that can also lead to cancer. Some pro apoptotic uh, factors are there which called mimetics. They can resist cell death that can lead to cancer. Maybe some aerobic glycolysis inhibitors are there that can leads to cancer. So these are the role key players that can lead to cancers. Okay. Then what kind of genes are damaged in cancer cells? What mainly the kind of genes that are damaged in cancer cells? One type is called oncogenes, that is onco means cancer causing, and there are some genes.
ম্যাম লাস্ট কি বললেন শুনতে পারিনি। ম্যাম
কি বলছো?
লাস্টে কি
শুনতে
বললেন শুনতে পারিনি। লাস্ট লাস্টে
লাস্ট
কি বললেন শুনতে পারিনি, কথাগুলো কেমন কেটে গেছে।
acha. What kinds of genes are damaged in cancer cells? Since cancer is related with the genetic changes, some genes are related with this cancer. So what are those different types of genes? Oncogenes. There are some genes that oncogenes, oncogenes, oncogenes. Oncogenes are the genes that directly can cause cancer. Okay. There are some genes that can suppress tumor, which is called tumor suppressor genes. One is example is P53. This genes resist the formation of tumor. That is why the name tumor suppressor genes. Tumor can be suppressed. Okay, that is why the name. The example is P53, BRCA2. These are all the names of genes. BRCA2 is mostly related with breast cancer. Okay, these genes, they prevent from cancer development or tumor development. Mostly P53 gene, it also plays some key role in G1's restriction point. Yesterday I have told you about P53, and this P53 protein or gene, it halts the cell division if it detects some damaged DNA. Okay, so that is why we call as P53 as guardian of the genome, because it helps in the protection. What do a guardian do? A protects child. Similarly, likewise, P53 also protects cell from getting, prevents cell from not getting into cancer. That is why it is called the guardian genome. Okay. So P53 tumor suppressor of the gene. It is also called the master regulator of diverse cellular processes. So this P53 tumor suppression gene, it regulates a different types of cellular processes in our body. What are those cellular processes? What are the main activities that P53 gene can do? It protects cell from replication stress. It protects cell from DNA damage. P53 can have can uh, protect cell from some viral infection by activating some oncogene. It protects the cell from hypoxia, that is, if the cell does not have enough oxygen. P53 can protect cell from some translational stress, which we all this are put as stress input. These are the factors, these are the processes which can put the cell into stress. So P53 helps in reducing this, and there are some other transcriptional output. There are some other genes along with this that can help, and that help in the process of that in concert with P53 P21 MDM2. These are names of other genes that can work, and that can help in this process, like cell cycle arrest, DNA repair, apoptosis, angiogenesis, autoregulation. So that's why this P53 is known as the master regulator of all diverse cellular processes. Okay. So coming to other theory of the theory of cancer, there are other theories like somatic mutation theory of cancer. This is not so important theory. In this theory, what we get that DNA damage is mostly caused by endogenous processes and by chemicals, physicals, and biological carcinogens. And due to this all chemicals and biological carcinogens, what happens, there is an accumulation of somatic mutation in the oncogenes and tumor suppressor genes, which ultimately leads to cancer. This is the straightforward somatic theory, somatic mutation theory of cancer. Then coming to the another theory, which is called stem cell division theory of cancer. Here we get that some due to some physiological, pathological, and environmental factors that promote the accumulation of cell division in stem cells by acting on the stem cell or on the stem cell environment, there is some accumulation of cell division in the stem cell, and that accumulation, and due to that accumulation, some reversible change can occur, some alteration can occur, which is in the DNA level or genetic level. And what is that? That is some accumulation of DNA alterations can occur, like mutation, or some other DNA alterations can occur, and due to this concerted or combined effect, we will get ultimately cancer. Okay. Then coming to theories of carcinogenesis, means the process of steps that is that takes place in the formation of cancer. These are not so important, but just remember the name. These theories consists of genetic theory, epigenetic theory, immunosurveillance theory, and monoclonal synthesis. Okay, these are the theories for carcinogenesis. You don't have it so much detail in your syllabus, so just I am mentioning the names of the theory for theories of carcinogenesis. Then carcinogens. What are carcinogens that are causing cancers? There are a lot of types of carcinogens, means the agents that are causing cancer. What are those? Some I've already mentioned. Some are chemical carcinogens, some are environmental radiations, some can be medical radiations, that is the radiations which are used in medical instruments. There are lots of high powerful radiations that are used during medical radiations, especially scanning, CT scan, all that instrumentations uh,
अच्छा
takes
कैसे हो?
place with the help of radiations. That can also cause, because the radiation is directly entering into the body, into our body, and in
कैसे
the
हो?
into and within our body there are lots of cells, tissues. Due to that, if someone gets a frequent exposure to that medical radiations, they can develop cancers. Some viruses can cause cancers. Some special types of viruses are there that can cause also cancer. Some other medications, some side effects of medicines that we are taking, then that leads to cancer. And the most important one is the lifestyle factors, how we are leading our life. That is nowadays the most important factor that is leading to cancer. Now, going to the steps of carcinogen, already told, there are lots of steps. There are lots. There are 4 steps. Number one, we call it as initiation, where cells we are seeing cells as a mass or lump of cells. Then, then the next step is the promotion, which is the which is the second step of the carcinogenesis process, and that promotion ultimately leads to progression and ultimately to to tumor or cancer progression. And here are given a list of carcinogens, maybe due to radiation. There will we get some free radicals, or we get some reactive oxygen species, and that reactive oxygen species can cause genetic aberration within our DNA. That is why we get damaged DNA due to chemical substance, or also like carcinogen metabolites that we get due to environmental exposure, that can also cause DNA damage, or damage can occur in the DNA due to virus, due to viral oncoprotein, viral genome. These can also cause damage to DNA. Due to bacteria also, some due to some bacterial infection or attack of bacteria, we get some pro-inflammatory chemokines and cytokines production, and that chemokines and cytokines that leads to chronic inflammation or development of ROS, and ultimately that induce DNA damage. Okay, this is how carcinogens are interfering within the damage of DNA, or in the genetic change, which ultimately cause mutation, and ultimately due to mutation we develop cancer. Okay, then coming to genotoxicity. What is genotoxicity? It's a special name given when there is toxicity lead to toxicity caused in the gene, that is called genetic toxicology or genotoxicity. That is mostly due to DNA damage, and if there is no repair occur, or if there is misrepair, the mutation can occur, and due to that mutation we can get the chromosome break, we can also get chromosome aberration, we can also get duplication of chromosome, that ultimately can cause cancer, which is also due to chromosome break, chromosome aberration, or duplication in the chromosome. What we get our genome as instable. Genomic instability will be developed, and due to that genomic instability, ultimately that leads to cancer in the long term. Then it's a very last slide, that how we can prevent cancer. Our cancer prevention recommendation, it is mostly given by World Cancer Research Fund and American Institute for Cancer Research. What are the lifestyles that we need to follow in order to prevent cancer, or some recommendations that we should follow or we should put in our lifestyle so that we don't get to this deadly disease. Okay, what are those recommendations? Those recommendations are a lot. We have we if we if we limit consumption of red meat and processed meat, if we limit consumption of sugar or sweetened drinks like the cold drinks, the beveraged drink, carbonated drinks, that if we can limit that consumption, if we limit alcohol consumption, and if we don't use some supplements that are present nowadays for cancer prevention, those supplements are not good for our normal activities of the body. They can interfere with our normal body cells, and that is not good for our body. So don't use any supplements for cancer prevention. For mothers, for baby, breastfeed your baby. That can lead to prevention of cancer. Okay, that is very much important. Babies also need mother's milk, and if mother is not uh, capable of breastfeeding her baby, ultimately what happens that the milk that gets deposited, and ultimately due to that deposition of milk, ultimately breast cancer can occur in the long term. Okay, then if starting from here, I have to start from, it's my fault. I have to start from here, because always be a healthy weight individual. Don't be overweight, and don't avoid obesity. That can lead to cancer. So always take a proper diet, proper nutrition to gain a healthy weight. Always be physically active. Always keeps the cells of your body active, reactive, so that your cells can work so that your cells can do all the metabolic activities that are going inside your body, that should be in a good level. Then eating a diet rich whole grains, vegetables, fruits and beans. Always eat nutritious vegetables, fruits, beans, antioxidants that can prevent your cell from stress. Always eat a healthy, always have a healthy nutrition and balanced diet in your diet chart. That is very important nowadays. Limit consumption of fast foods and other processed foods that are high in fat, starches, or sugar or trans fat. Like the fast foods, like the burgers, pizzas, that contains a lot of fat in form of cheese, that is very unhealthy for our health. And then everything I have told, the limit consumption of red meat, because red meat is not good for our health. Then consumption of sugar sweetened, limit consumption of alcohol, supplement breastfeeding. And I think if everyone follows all of this more or less, we everyone can prevent cancer and can lead a healthy life. Okay, so that's all for your cell biology syllabus. Here we mostly end our cell biology syllabus. Okay, so any question? হ্যাঁ বলো। Molecular basis of cancer. There are, yeah, in the notes I have given you. As far as I remember, yes, that is how cancer is related to molecular biology. events. That may okay, that I will discuss you in January. Just be clear with the basic concept, and then then some harder things I will discuss with you in the month of January. Okay, that is the interaction with cancer along with molecular biology. What are the molecular events that are related to cancer? Okay, characteristics of some molecular biology related to cancer. That, হ্যাঁ বলো। হুম। Yes, because Lellinger is mostly a book of biochemistry. Cancer you will mostly get in cell biology book. In some parts in genetics, some genetic book, you can get some part of cancer there. Because since cancer is related with genetic changes and everything, but in biochemistry book, you won't get such details of cancer there. Okay. So cancer is a very specialized subject. It's a what I say, combination of many subjects, cell biology, genetics, developmental biology, everything. So actually, this cancer is a very high level type of subject, and high level uh, thinking, and high level everything high in occurring in the in almost in all level. So it's very, in this stage, in this preliminary stage, it's very tough to grasp the whole thing about cancer. Whatever I try to explain in a very simpler way in the slide, and try to cover on almost all things that are included in cancer. So, and you just go through the slide, and if you feel to read more, and if you feel to know more about cancer, then you can go through some through some research journal or review papers on cancer. So there are lots of things about cancer, because cancer research is going on for a longer time, and till it's going on in future, maybe it will go on. So it's very tough to what I say, in a nut to make cancer in a nutshell thing. That is the what I say, the limitations about cancer or oncology. It's so vast and it's so interrelated, so it's very difficult to make you understand all things. So it is related with molecular biology, it is related with developmental biology, it is related with genetics, everything. So হ্যাঁ বলো। Yeah, if you are always encouraged to read, but that I think will feel somewhat complicated in Albert's also. What you can do, let me check if I can get some review papers on cancer. There are lots of research journal review papers on cancer. If I get if I get uh, at your stage, I will post it on your Google Classroom. Okay. Okay. If you find it easier, you can go through. You are always encouraged to go through. You can read CARP also. CARP, if you have CARP, if you can download CARP, Gerald CARP, you can read it from there also. Gerald CARP is also a good book of cell biology. Okay. Yeah, much information you can't get because it's still a research level subject. If so many research is going on cancer in so many ways, in so many direction, it's very hard to grasp that all things. That is this cancer biology is always a very mysterious thing. It was a mysterious thing, it is a mysterious thing, and it will be a mysterious thing actually subject. There are lots of theories that they are related to cancer, lots of theory. So it's not possible at your stage to cover all. So and you are not able to understand in that level because it's it's only your first semester of your B.sc. So that is why it's very tough. Okay, anything? আর কিছু কারো জিজ্ঞাসা আছে? Gerald Karp. Gerald Karp. Cell Biology by Gerald Karp. Karp is a good book. Not you will also get other chapters there also. Some biochemistry things also you get. Yes, Gerald Karp. Signal transduction, apoptosis, everything is there in that book. Also, some biochemistry portion is also there. And yesterday I uploaded one book that is very easy book if that will cover mostly all aspects of your biotechnology subjects. You can in the first stage you can see that. Yesterday I have uploaded that and uploaded that in the Google Classroom. As a preliminary thing, you can see that book also. It consists of molecular biology, some genetic portion, some biochemistry portion, some cell biology portion, some molecular laboratory techniques are also there, which you will encounter in your coming semesters. So that book you can check. Okay. Indian books you can also go. Rastogi, there is Cell Biology by Rastogi. But I don't prefer Indian books because mostly Indian books are copy of the international books. That's why it's better, good to consult international books. Okay. আর কিছু কি আছে জিজ্ঞাসা? তাহলে আই উইল বি সাইনিং অফ। ওকে, আর একটা কথা জিজ্ঞেস করছি, তোমাদের কি প্র্যাকটিক্যালটা আমি ওই ডিসেম্বরের 27th স্টার্ট করব, ঠিক আছে? তো আমি কি তোমাদের একটা আমার পিপিটি করা আছে যেখানে দেয়ার আর লিঙ্কস অফ দ্য ভিডিওস অফ ইউর প্র্যাকটিক্যালস দ্যাট আর গিভেন ইন ইউর সিলেবাস অফ সেল বায়োলজি প্র্যাকটিক্যাল, আমি কি সেটা আপলোড করে দেবো? তোমরা এই ব্রেকের সময় তোমরা তাহলে দেখে রেখো। ওকে ওকে, তাহলে আমি আপলোড করে দেবো। দেন আই উইল প্র্যাকটিক্যালে তোমরা দেখো এই ব্রেকের সময়, তারপর আমি থিওরি রিলেটেড টু দ্যাট প্র্যাকটিক্যাল কি কি আছে আমি বুঝিয়ে দেবো। ওকে
ওকে ম্যাম।
ওকে ওকে, ঠিক আছে, বাই। অ্যান্ড স্টাডি আ লট, study during this break so that you can we can do a good revision that will start from January. Okay okay, and also during this break go through your PPT presentations. Then at the end of December I will give you the date of your presentation. Okay, maybe okay. Okay. Thank you. Okay, thank you.
Thank you, ma'am.
Best of luck, ma'am.
Ma'am, all the best.
Thank you. Okay.